little bit late, so unfortunately we have to skip the break and go directly to the next talk. And it is a pleasure to give the floor to Dr. Cristina Gardelli. She is Associate Director in Medicinal Chemistry, Research and Early Development sector in the Division of Respiratory and Immunology of AstraZeneca in Gothenburg in Sweden.
Okay, thank you very much. Professor Saso, can you all see my slides?
Yes, very well.
Yes, thank you very much. So I would like to thank Professor Saso and all the organizing committee for the invitation to give this webinar to the UNICA Students Network. And it's my pleasure to show you some opportunities in the drug discovery area through a big company such as AstraZeneca, that is the company where I have been working for the last 10.5 years. AstraZeneca is a big pharmaceutical company with more than 24 billion total revenue, and we invest 6.1 billion in R&D. We research across 6 countries. We had 28 new medical entity approved in 2019, and it's a company with more than 70,000 employees worldwide. I belong to the medicinal chemistry department in the respiratory and immunology function as a team leader and with project leader responsibility at the Gothenburg site here in the picture, so in Sweden. This is one of the biggest R&D sites from AstraZeneca with 2,400 employees. Of those, 2,000 are scientists from 50 different countries, and among those, 600 have a PhD, and 30 are involved in academic roles as professors. At Gothenburg, we cover all the chain from target biology up to clinical development. And recently, we have split it actually, the organization into we have split the R&D into organization, oncology and biopharmaceutical, to increase focus in this, our therapy areas, and also to have a more agile decision-making and resource allocation process. So we have oncology R&D, where we have work in several different types of cancer, like lung, breast, pancreatic, liver, prostate. And then we have the biopharmaceutical with cardiovascular, renal and metabolic function, respiratory and immunology, and then neuroscience and microbial science. These 2 organizations are very well connected in terms of collaboration, in terms of business. Why? Because we have a lot, sorry, I needed to minimize. We have a lot of functions that are shared between the 2 organizations. And here you see that when we do research, as it was said by the speakers before me, of course, we need people that have experience in many different disciplines: discovery science, antibody discovery, clinical pharmacology, pharmaceutical science, biopharmaceutical development, in digital health, data science, precision medicine and biomarkers, or then you can be involved in business development, licensing, and so on. I will focus my presentation mainly in the drug discovery part, that is a really early phase of discovery of a drug. And if I'm almost around 25 years of experience working in biotech or big pharma. And if we look at the overall journey from target identification up to patients, in a micro way, let's say, this has not changed so much. We go through target identification, then hit and lead generation, lead optimization, all the study in preclinical, and then the clinical trials, and then we reach the market and the patients. But what has been changed a lot in these 25 years are our mates that work with, at the beginning, many years ago, the core team was just the biologist, the chemist, a DMPK, a person with expertise in toxicology, and the clinician. But now we work in a much, much more multidisciplinary environment. So we need people that have information on omics, for example, and this omics area gets bigger and bigger every day. Genomics, transcriptomics, proteomics, metabolomics, and so on. We don't deal anymore only with small molecule or with peptide or monoclonal antibody, but we have a much richer toolbox of new modalities. We need people that have experience in automation. We need people that have experience in silico prediction, in modeling and simulation. We need people that have experience in mass imaging, in digitalization. We need biomedical engineering because we want to study the organs on the chips. We need people that have expertise in precision medicine, and so we are aiming to personalize healthcare. I said that we need people that have experience in omics because omics is really the projection of the high-dimensional biological space. So I put just an example of a biological event, but when we look at a biological event in early phase in drug discovery, we can look at it with different eyes. And we have many different technologies that allowed us to look at the phenomenon with a different perspectives. For example, through NMR, through mass spectrometry, through RNA sequencing. And then, from all the analysis of this data, we have very good information about the metabolite of our drug, about proteomics, so to understand how the protein interact each other, or about the transcriptomics that allowed us really to understand better the disease mechanism and also if the therapy is effective. So we use all this big data to identify new targets, but also to understand the mechanism of action, also to detect on-target or drug-induced side effects. And I'm a chemist as a background, and nowadays in the medicinal chemistry department or in the antibody and protein engineering department, we are aiming to build a much stronger toolbox of modality where we have small molecule, where we have PROTAC, where we have monoclonal antibody, where we have antibody drug conjugate, bispecific antibody, anti-caling. So you can see that this toolbox is much, much richer than it was in the past. And we need people that maybe started with chemistry studies, but then they specialize themselves towards one specific modality, people that have experience in oligonucleotide or people that have experience in peptide or in mRNA and so on. In this case, we are able to attach targets that in the past were considered to be undruggable. And when we build this, we look not only to have more modalities to attack new targets, so to attack new disease, but also try to see if we can have a specific targeting to a specific tissue, to a specific cell, to a specific organ. Of course, I said that we need people that have also expertise or that have passion, I would say, in automation. The automation in the labs entered many years ago, I would say already 25 years ago, with the combinatorial chemistry. There was a lot of automation in our lab. But now we are talking more of digital laboratories, where the automated synthesis, where the analysis workflows are very well integrated and maybe are able to work 24/7. We wanted to integrate also all the technology to purify the compound, to elucidate the structure of this compound, to make quantitative samples. Then we wanted to integrate all the assay about the biochemical, biophysical, compound properties. And ultimately, or I would say at the base, we want to connect the drug design with the AI component. in order to make this cycle, that is more or less what we do in the very early phase of drug discovery, the cycle design, make, test, analyze, much faster, much more quicker. So I would say the importance of artificial intelligence and automation, and this was cited also by Professor Thomas and before. This is what is really, I would say, the big innovation in our labs nowadays. And also, if we are a little bit, at the beginning of apply artificial intelligence in drug discovery, we have many more projects nowadays in which the traditional medicinal chemistry goes in parallel with the artificial intelligence approach. This will have definitely a big impact in all phases of drug discovery, is already having this, and I just reported some paper for you if you are interested in this, where in the first paper from Nogradi, the scientists in 2019, this is a pretty general paper, but it really shows this example with terbinafine, the discovery of the terbinafine metabolites with an artificial intelligence approach. And then we rely on artificial intelligence for some of the most important challenging of nowadays. For example, if we think about the discovery of new antibiotics in order to, solve this antibiotic resistant problem that is really a current big issue of the humanity, I would say. So if artificial intelligence can help us to design the next generation of antibiotics, can help us in predict the impact on health and to have a more personalized and precision medicine approach. And artificial intelligence needs to help us in having faster and safer clinical trials for most of our disease. So when I say that we have new trends now in our labs, I'm referring, for example, to all the new cellular, humanoids, organoid models that really help us to bridge this gap between animals and humans. We are using more and more the molecular imaging, and this is very important because molecular imaging allows us to investigate what happens in the three-dimensional space of a cellular. So we are able to study not only in a more physiological, through environment, the disease, but also if our therapy is really effective or not, with the analysis and the quantitative measurement of all the biomarkers, for example. So these are just a few slides to show you that, and this is something that is very, very close to my heart. When you shape your studies course, think about what will be necessary in the next 10, 15 years. And technologies that now are really at the beginning, they will be really the base of the research, of the development in 5, 10, 15 years. And so you need to keep in mind what is new now in order to be prepared to work in the pharmaceutical environment in the future. And there is the course of study at the university, but there are a lot of tools that you can use in order to complement your background. You don't need to spend an entire course of study in artificial intelligence, but you need to understand a little bit what is an algorithm and where we could apply this. So think about, for example, if you wanted to have extra exams or an extra study in order to understand a little bit what is an MR, what is mass spectrometry. Also, then you don't become an expert, but you need to understand in the discussion all about these physical methods and so on, if you want to stay in a multidisciplinary team at the very early phase of the drug discovery. Then I've been part of other events with some of the colleagues that are part of this seminar in the past, and I wanted to use this occasion to present our research and development graduate program, because this is a way to enter in the pharmaceutical company. You know, many companies have this graduate program, but AstraZeneca has this in particular that was launched in 2013. And is a way to enter in a company with just a master degree and to be able to explore what happens in a pharmaceutical company. This is a 2-year program with 3 different 8 months placement across R&D. And this program is run in Europe at Gothenburg and Cambridge in the UK. And this is an excellent opportunity to explore different areas of the drug discovery process, because the candidate can shape his journey. I can tell you that we are now approaching the third rotation of the cohort from 2018, the last rotation for them. And we have 8 graduates, and the scientists at our site offer 145 vacancies. So a huge offer, many different things. And you can spend 8 months in one department that can be more or less in the same area. For example, there are chemists that want to spend 8 months in synthesis, 8 months in computational chemist, 8 months in analytical. But there are really people that want to use this opportunity to explore areas of the drug discovery process that is unique to a big company. So maybe they start with a placement in chemistry, then they want to go in the MPK, they maybe end up and doing a rotation in the clinical department. With this, we have also the enrollment in a global graduate development program, and this is very important because you need to develop also your softer skills together with your hardcore skills, and then you will have a mentor that follow you for 2 years and a competitive salary and benefit. You can apply for the next cohort if you graduate this year or your graduation is due in 2021. And the requirements are usually, you know, we want people that are really interested, that have a real passion for drug discovery and drug development. They like to be innovative, they have strong critical thinking with a lot of enthusiasm and energy. And this is the link that you can use to apply for our original R&D graduate program. But since we launched this program, this has been very successful. Everybody wants to have a graduate because, you know, these are people that are very motivated, and for them is a big, big opportunity. And they want to get the most of this. Then they decided, some of them apply for position in AstraZeneca, some of them go and work in other companies, some of them go and make the PhD with much more awareness of the field in which they would like to run a PhD. That's why we have created program also in other areas, not only our classical R&D, but we have this 2 years program also for biometrics and information sciences, for biopharmaceutical development, data science and artificial intelligence, for people that maybe they studied you know, science, but then they realize that this is not really what they are passionate about. They can also have a big opportunities in big pharmaceutical company working in other area like human resources, for example, IT, technology leadership, and we have done a program in operations, supply chain, and PT&D, pharmaceutical technology and development. How to apply is very easy. You go to this careersastrazeneca.com students, and nowadays we have become a little bit more innovative. So there is a video interview. For some, there are some online games that you need to play. And then if you are selected, you will be called for an assessment center. Maybe this year, next year will be virtual. Hopefully not, but usually people are called here on site, and they spend an entire day here on site. And then if you are successful, you will receive an offer of employment. One thing that I wanted to mention, because I'm part of the Great Place to Work task force, when we apply for a job, we need to match our desire, our expertise, but we have to be also careful to watch the values of the company for which we work are. And I'm very proud to say that, in our company, 87% of the employees agree that their manager supports diversity and inclusion. And this is very important. Empowering diversity is embedded in everything we do. We have 45% of our senior roles that are filled by women. We, from the last survey, 87% of employees have improved their skills, learned new skills, or had a development opportunity in the last 12 months. I really liked what was said before also by Joseph and by Andrea, that if you want to work in a pharmaceutical company nowadays, you really need to be a long life learner, because you start with some skills, but you need to develop a new skill, hard and soft, every day. And this is something that we don't need what has been taught, only what has been taught, but we need your attitude to learn new things every day. 61%, so we are really committed to sustainability. 61% of our total electricity use is sourced from renewable sources. And then it was mentioned also from Professor Thomas, it's important that we work with a good life-work balance. So 65% of our sites have also programs to address employee well-being. So this is my major message. I presented the graduate program, but if you go into astrazeneca.career.com, of course, you find also a permanent position for all your interests and so on. But don't narrow your field. You can look at opportunity close where you live, and also, in other countries and so on, because it's important that we keep building knowledge, building expertise. And then maybe we have opportunity to go back to our own country, or we will enjoy to stay where we end up. So be open and don't close your, broad of exploration, your area of exploration and so on. There are opportunities, and so we need just to keep up the passion and the motivation to look around also. And with these things, I want to thank some people, because I borrow from them some very nice, colorful slides, and are all people that are working hard in this opportunity for young scientists. And then also my head of department, Vanga Cecisi. And thank you all for your attention. And if you want to contact me for having more information, please do that. I'm sure that this package of slides, Professor Saso, will be made available now for the entire network. And
So
thank you very much all.
Thank you very much, Cristina. Thank you very much. Really was a wonderful presentation, very useful, very concrete, also optimistic at the end. I also agree that we have to be proactive. You are a very good example of a person that faced different challenges in a very good way, and now is working in Sweden. And I fully believe that especially mobility within Europe is not really going abroad. If we really believe in our European Union, I think the mobility within the Union should be allowed and encouraged. So thank you very much. And it is a little bit late. Thanks. It is a little bit late, but this is due to the passion of our speakers. I think all of them really want to share with you their experience, they provide useful information to the participants. So I hope you will be patient. We will try to close in about 40-45 minutes. So I really ask the next speakers to keep the time. And now it is a pleasure to give the floor to Dr. Glyn Steventon. He has been a friend for a long time. We met actually also in another country. We met in Madrid in another occasion. So he's not originally part of the photograph that Professor van Schavedijk actually projected, but is another very good relationship that we kept for many years. And now due to COVID, we managed actually to really we establish by Zoom and other online meetings. Is an expert in pharmacokinetics. He has been working in several organizations, and at the moment is working at the Defense Science and Technology Laboratory in the UK. And his division is a chemical, biological, radiological division in the group of toxicology, trauma, and medicine. So he really has a very good, experience in positions related to pharmacokinetic studies, and he will give a presentation on that. Thank you, Glyn.
Thank you, Luciano. Hopefully you can all see the screen there.
Perfect, yes.
Right. For the students who've stuck with us all the way through this really interesting morning, I am not going to torture you with equations and mathematical models. Yes, this is what pharmacokinetics is about to some extent, but it is about much more than this. It's about how you actually take the whole process of ADME, absorption, distribution, metabolism, and excretion, and actually help to design later stage trials and also take data from the preclinical development studies to help these things to be seen. So what we're talking about here is pharmacokinetics, pharmacodynamics, and to go with what a previous speaker has said, it's now starting to be called pharmacometrics. And the questions we're trying to answer here is, which subjects are we going to include in our later trials? Is because of the preclinical ADME studies, is there a reason to exclude individuals from a clinical trial? Or are the reasons that we're actually including individuals in a clinical trial? What is the first dose into human? Which is going to be the best design of the clinical trial to show us proof of concept? And also, what is the dose going to be for efficacy? Some people not only call this pharmacometrics, they actually also talk about model-informed drug development. And what we're trying to do here is take information about the disease process, the drug itself, the new chemical entity, the new biological entity, and also the clinical trials to see where we're going. I'm sorry about this. This is a downside of working for a government body. The opinions expressed in this presentation are solely my own and do not reflect the view of DSTL, the Ministry of Defence in the UK, or the government of the United Kingdom of Great Britain and Northern Ireland. So I apologise for that. There's my disclaimer. So you can blame me for everything and not take anybody else to court over it. So preclinical pharmacokinetics, what is it about? Well, in silico, well, clearly in silico. But let's be honest, we're all working in silico at the moment, courtesy of COVID, but there are other aspects to it. It is just not about mathematical modeling of plasma concentration versus time. There could be prediction of major sites of metabolism. So Cristina's computational chemist could actually pass a molecule over, which has not been synthesized yet, and it then goes into a platform where, known about the physical chemical properties of the structure will be, we can predict what the major sites of metabolism are. It doesn't necessarily mean that we don't need to do metabolism studies. And on top of this, there are also other platforms where you can predict whether the compound is going to be absorbed. If you've got very low bioavailability, that could be a problem. And therefore, based on this, some discussions could take place. It could then, therefore, I end up having a meeting with Cristina, and Cristina then changes the structure of the compound to get around these problems. So computer modeling is really important. Pharmacokinetic modeling. Most pharmacology or pharmacy students now have probably been tortured with semi-log graph paper, either online or actually in a paper version, and the equations where they've had to model out plasma concentration versus time. the biotechnology students and the biochemistry students have probably been tortured by their lecturers where they've made them do enzyme kinetics, determine Km and Vmax. And Km and Vmax are important because they can give me clearance through an enzyme. And based on clearance through the enzyme, I can model that up to clearance from a body, whether that is a mouse, a rat, a dog, a non-human primate, or a human. So pharmacokinetic modeling is important. And again, some of that is in silico, but also it is also based on laboratory derived data. And this can come in two forms. It's either a non-compartment model where we just actually look at the whole body as a single compartment, or a more complex version of it, which is called compartment modeling. In this case, we're looking at the interrelationship and the movement of the drug from various compartments. So the central compartment would be the blood, and then you would look at how it moves from the blood into, say, the liver, which could be one compartment. The kidneys could be a second compartment. The central nervous system, the lungs, other compartments, but it doesn't necessarily have to be an organ. Adipose tissue, either brown fat or white fat, could be a compartment. The bone could be a compartment. Muscle could be a compartment. For the pharmacologists and possibly the biochemists also, that is the pharmacodynamic modeling. This case where we're actually looking at the effect of the drug on the body. And also we can actually do other things in terms of pharmacokinetic predictions using physiologically based pharmacokinetic modeling. And what I'm going to try and do is show you what these things look like here. Whoops, I'll apologize for that. There we go. So pharmacokinetic modeling, what we're looking at is changing the dose of the drug and seeing what effect that has on concentration versus time. Pharmacodynamic modeling, which is where the biochemists and the pharmacologists may come in, where now we're changing concentration, but we're looking at the effect this is actually having on the body. And then finally, we can combine the two together, where we're actually looking at the changing of the dose and seeing what effect that has on concentration versus time. Now, invariably, these use quite complex software programs, but my attitude has always been when I've spoken to students about PK, PD, PK-PD modeling, is if you can add, multiply, subtract, and divide with a calculator, you can do PK. Certainly when you get your career going in pharmaceutical industry, the biotechnology industry, or the regulatory agencies, you're not going to be dealing with a calculator and a pen and paper. You will be doing things through a software program, but the principles are the same. And what we're looking for is people being able to see a pattern and understand what that pattern means. So non-clinical pharmacokinetics. Let's try and put it into a visual picture in terms of what the previous speakers have been talking about. So here is your design and synthesis of drug, and going through all the processes lead optimization to putting the drug on the market. There is your synthetic chemistry, there is your formulation sciences, there is your production science, and it all interrelates and confers back into this cycle. So there may well be initially an in silico process where the in silico chemistry talk to the in silico PK, the in silico drug metabolism, and the in silico toxicology. But at some point, somebody's going to manufacture, synthesize a new chemical entity, a new biological entity. And then the studies actually start. And the studies become quite simplistic. You may actually be looking at protein binding. Does the compound bind to plasma protein? Does the compound bind to brain protein? You may also therefore need to look at permeability. Does the compound become absorbed? Now for biologics, this is not a problem because they don't tend to be taken orally. But for a new chemical entity, a new drug entity, the major route of administration is orally. And if you can't get the drug from the GI tract into the bloodstream, that is a problem. And this will be done through in vitro testing. Likewise, your test systems, you go from proteins, cells, to an actual animal. In this case, it may well be that your efficacy studies have been done in a mouse, and it may well be that because it's developed to treat a disease, you've got either a knock-in or a knockout mouse. And what I mean by that is that a gene has been knocked out which causes a disease, or a gene has been activated or inserted that causes a disease process. And therefore, you're going to try your compound to see if it actually ameliorates the disease, stops the progression of the disease, or changes other factors. But likewise, further on, you're going to have your toxicology studies. Invariably, your toxicology studies and your preclinical pharmacokinetics will take place in two species. In this case, we're talking about a small rodent, mouse, rat, and then a larger mammal, in which case I've actually shown a dog. But it doesn't necessarily have to be a dog. It could be a non-human primate, or it could be a mini pig. And the object of the exercise there is to start to tie in your preclinical in vitro studies with your preclinical in vivo studies. So you would also move on and look at metabolism, and invariably you would look at this in vitro. You would look at the liver because it's the major site of metabolism. You would break that down into subcellular fractions, and you would look at the S9. So the S9 is the endoplasmic reticulum and the cytosol fraction combined, which has where we call our phase 1 and phase 2 reactions. Or you would just look at the microsomes, the endoplasmic reticulum, where the major phase 1 enzymes, and you will be looking for what is the metabolism. Is it metabolically stable? Does the compound degrade either chemically or does it degrade metabolically? If it undergoes metabolism, what's the extent of metabolism? If the metabolism is greater than 10% for a particular metabolite, you will need to actually look at the studies involving those. Invariably, these will involve analytical chemistry, hence the little diagram there for the LCMS-MS. This will feed back into everything. You will get metabolite identification from this. Out of your metabolism studies, you'll get an idea of possible drug-drug interactions, or whether there is going to be pharmacogenetic problems. There are people who actually have defects in drug metabolism enzymes, and you may want to actually avoid a drug going down that metabolic route. You'll do KM and Vmax determinations, and they again will then feed up into your modeling. At some point, you're going to take all of this data and put it together, because you're going to go first into human. And in that case, you're taking all your efficacy work preclinically, your DMPK preclinically, your chemistry, and you're putting it together to be able to come up with a dose that is going to be safe in the first into human. From there, you could easily move on into population pharmacokinetics. You can see PK studies done during clinical trials, and it may well be that you're actually doing a PK study within a target population, or it may well be it's a special population. Certainly for me now, I'm probably getting closer to the geriatrics population than I am to the pediatric population. But DMPK will take place across all these areas. The diagram that popped up onto the right, I've deliberately left it up there. I've not talked about it yet. These are all the things that you will probably do during your non-clinical pharmacokinetics and drug metabolism. And if I put it into the context here, all companies will do these studies, but not necessarily in the order stated here. Companies have their own way of doing things. In the end, they will do these studies, but they may not be in the order that I've suggested. And that simply shows you that not all roads lead to Rome, but they do eventually. It's a simple case of how you get there. And it may well be that if you're dealing with a new chemical entity, you do an awful lot of these things. If you're dealing with a new biological entity, then the metabolism side is actually very poorly followed through. Why? Because most biological entities are degraded back into the constituent amino acids. They don't have active metabolites. And because of that, you don't need to do these studies. So that is your sort of non-clinical pharmacokinetics put together in terms of what's going on, in terms of visualizing what the previous speakers have already said. But don't take my word for it. previous speakers just mentioned, there are numerous things. You could go onto the AstraZeneca website, you can go onto the Pfizer website, and they'll tell you all these things and what they do. But the CROs give me a little bit of a more adventure. So here are 3 CRO companies, not that I actually are recommending them in any way. I deliberately picked them for their geographical basis. So Cyprotex at the top was a UK-based company, which has now been taken over by a US company. ADME is a Finnish company who has just been taken over by a Dutch company, and Covance is a very large US-based company, but they're around the world. And if you go to these websites, these addresses were live this morning and were easily accessible. You can actually see the studies they offer, not only to small companies, but to big companies as well, in terms of preclinical ADME and preclinical pharmacokinetics. Universities do them. It depends where you are and in which country, but most universities around the world actually have some ADME PK in some form. Here, the University of Manchester, it has a consortium with 9 of the top pharmaceutical companies, and they are probably the premier site for PK in academia in the UK. And then finally, you've got the modelers. Well, then Certara is probably the biggest of these companies. They're not exactly a modeling company. They will do modeling for you, but what they do are the home of 2 of the big platforms for carrying out pharmacokinetic studies. The Phoenix PK/PD and the SimSip PB/PK simulator software platforms. These are used by at least the top 100 pharma companies, and also the US Food and Drug Administration and the European Medicines Agency. So these platforms are available. I use the Phoenix PK/PD software myself. We don't use SimSip at DSTL because our portfolio of compounds is a lot smaller than a regular pharmaceutical company. But there you can see that everything the speakers have been talking to you about are used by companies, big or small. What do non-clinical pharmacology, pharmacokinetic studies require? They require you. We want graduates in analytical chemistry. We want graduates in biology. We want graduates in biochemistry. We want graduates in pharmacy, biotechnology, mathematics. Without graduates in these study areas, you are, or we are, completely lost. We have no input into our new intake of workers, and we have no ability to force and the drugs through their programs without graduates. So if you're sitting there thinking, well, what I do isn't really applicable to the pharmaceutical industries, I would turn around and say what you do is highly applicable to the pharmaceutical industries. I would take on board what some of the previous speakers have said, certainly from the students. Sometimes you get taught things at university which don't appear to be in the context of what industry want, but I would point out to you that the vast majority of what you learn as an undergraduate is what industry want. It's just that industry needs to train you up and educate you to think about it in a different way. So hopefully I've got us slightly back on time, Luciano. I'd like to say
yes, perfect.
Thank you to the previous speakers for setting this up, but also thank you to the students for listening.
Thank you very much, Glyn. Very interesting also for opening this door towards some positions which are not very much known, among students and the recent graduates. So I think that was very useful. And now, last but not least, it is a pleasure to give the floor to Dr. Mark Martens. He's a former Vice President of Nonclinical Development of Biotech, the Johnson & Johnson company, and consultant in preclinical development and toxicology. And he will, again, talk about career opportunities in non-clinical development, but in this case, with an angle of related to toxicological studies.
Thank you, Luciano. Thank you very much for inviting me to take part in this seminar. It's quite interesting, and I'm actually proud to be able to share my experiences in chemical and pharmaceutical industry with the students in Europe. My talk will consist of 4 parts, a very short introduction, which I will keep very short because already a lot has been said on this subject. Then management structures in non-clinical drug development, and this more in regard to toxicology, career paths in non-clinical drug development, and then tips for a successful career in the pharmaceutical industry. So you've seen that already several times, so that is the drug life cycle. I won't expand on this. The only thing I would like to point to you is at the bottom of the graph where you see the intensity by which non-clinical development is involved in drug development. So the most intense activity in most projects were handled by non-clinical development at the phase of early development, and then later, taken to the pre-approval late development. So a lot has been said about the content, about the type of skills that are needed to be able to contribute to drug development. But now I'm going to talk about the type of management structures that are required in order to forward the drug from a drug discovery up to marketing. Now, since drug development is a multidisciplinary activity, a lot of people with different skills and different educational backgrounds are contributing to the process. And if you start with the level of early development, and then later also late development, at the level of drug development, a team is formed where several disciplines have to contribute. And in this case, for the drug development team in early development, you will have the contribution of discovery, because of course they are transferring the drug from discovery into early development. There are contributions from clinical pharmacology and medical, clinical operations, regulatory, finance, project planning, chemical and pharmaceutical development, clinical pharmacokinetics, and non-clinical development. So non-clinical development is only one party who will be contributing to the whole process of drug development. Now let's concentrate on non-clinical development. So the non-clinical development team leader is the person who actually attends to the meetings of the early development drug development team, but in turn he will have to, or she will have to, chair the meetings of the non-clinical development team. Now that team, of course, is also composed of people with a lot of different disciplines, of course, like toxicology, safety pharmacology, project business and business management, quality management, pharmacokinetics and toxicokinetics, and regulatory document management. So the non-clinical development team leader collects all those data, integrates the data, and represents the results of non-clinical development to the whole drug development team. Now let's home into toxicology. So the toxicology leader who sits on the non-clinical development team in turn has to be connected with the laboratories where he has to place all the studies that are required in order to get a full dossier to pass from the one development phase to the other. So we call him the compound toxicologist. Now, a compound toxicologists from most of the companies these days, they place their studies either in their own laboratories, which is becoming more and more rare, but most of the time into contract research laboratories. And there he's in continuous contact with the toxicology study director. And actually for each toxicology assay that has to be placed, a toxicology study director is appointed by the contract research laboratory. Now, if you go up to the level of the contract research laboratory, the toxicology study director coordinates all the activities that are necessary to come to a complete toxicology report that has to be submitted to the compound toxicologist, who in turn will integrate all the toxicology studies, make a digest, and present a risk analysis to the non-clinical development team. Now, what are the disciplines that are contributing to the work of a toxicology study director in the laboratory? That's biostatistics, laboratory animal science medicine, bioanalysis, project business management, formulation chemistry, laboratory quality management, necropsy and histopathology, clinical biology, and laboratory management. So all those disciplines are needed by the toxicology study director to be able to present a report to the compound toxicologist, who will take it further on to the corporate level and to contribute to further drug development. Now, why am I explaining this type of managerial schemes? These are based more or less on my own experience, and I'm sure that other companies may have been organized or organized in a slightly different way, but generally it comes down to teamwork where several scientific disciplines are contributing. I'm using these managerial schemes to actually allow you to to draw a couple of career paths that are possible throughout the drug development process. Now, what kind of diplomas, what kind of degrees are needed to realize drug development in the non-clinical field? These are biomedical sciences, biology, molecular biology, biotechnology, pharmacy, veterinary medicine, chemistry, bioengineering, physics, and statistics or mathematics. Okay, well, let's look at a couple of possibilities of career paths that can be followed in pharmaceutical industry. And let's start with the laboratory level, because when you are graduated from pharmacy school or from degrees of biotechnology, you will have to start somewhere. And I must, admit that in industry, priority is given to pharmacy or biotechnology degrees who already have some postgraduate experience in terms of a PhD, in terms of postgraduate specializations. In order to be able to already be sufficiently skillful, start somewhere at the laboratory level. The best way on how to start for toxicology and non-clinical development in general is to start at the laboratory level, where you get acquainted with the way of organization and the skills that are needed in order to produce results of high quality that can be used into submission reports for drug registration. So in this scheme, let's look first at where are the opportunities for pharmaceutical PhDs. If you look here at the bottom, well, the most logic entry for pharmaceutical PhDs could be formulation technology, or could be also bioanalysis. Bioanalysis for the analysis of drugs and proteins, small molecules, and biological entities by means of all kinds of analytical techniques. In order to actually be able to start a career at that level, you already need the PhD level where you have proven actually to design and master a project of bioanalysis. But if you are spending some time at the level of bioanalysis, then in that case, there are possibilities actually to move into department management or the bioanalytical management, because all those disciplines, they are normally, are part of different, management departments and have their different managements. So you can move from bioanalysis to either management if you like to move into the management career, but a very logic step for a bioanalysis is actually to specialize yourself into vitro metabolism, move further into non-clinical pharmacokinetics, and why not, then actually transfer to a different competency stream in the development process, for example, clinical development, where you can actually grow into a role of clinical pharmacokinetics and further, grow into that direction. There is also the possibilities of actually from non-clinical pharmacokinetics also to grow into the function of a pharmacokinetics study director. We are taking on all the studies in the laboratory in terms of ADME, absorption, metabolism, excretion and distribution, but also that allows you to grow into a more corporate role, which I will show you later. The in vivo toxicology, most of the time, the type of people entering that function are in the first place actually doctors in veterinary medicines, but it's also open for PhDs in biology and medical biomedicine. But that depends on the experience that you gained in your PhD on handling animals and this laboratory technology, etc. For in vitro toxicology, that is also a port of entry for pharmaceutical PhDs. Quite a lot of faculties of pharmacy have specialties in vitro toxicology, where all those new technologies like omics, etc., are acquired, and that is very important in order to start a career at the laboratory level in vitro toxicology. But once you get one moment in time saturated of in vitro toxicology, you may move into in vivo toxicology so that you can combine both skills, which is an excellent stepping up for a toxicology study director or a compound toxicologist at the corporate level. Now, if we move up to the corporate level, that means that is the level where actually the drug development teams gather and more or less produce the documents that are needed for registration. So when I go back to the toxicology study director from the laboratory, and having been in charge of the coordination of several toxicology programs, several types of toxicologies, like, for example, repeated dose toxicology, inhalation toxicology, reproductive toxicology, etc., then you have a very good and solid experience basis in order to become a compound toxicologist, contributing to a non-clinical development team as the toxicologist. Now, whilst being in a non-clinical development team, then you get acquainted with all the disciplines that are contributing to that non-clinical development team, like compound pharmaceuticals, but also clinical development, etc. And there are possibilities to grow into an early development team leader. And then why not, if you are getting completely skillful in early development team leader, you can then move on to become a late development team leader, and then even grow into portfolio management of even higher in the organization. So there are a lot of possibilities of career paths throughout pharmaceutical development. For example, I mentioned pharmaceutical PhDs who could grow into pharmacokinetics and compound pharmacokinetics, etc., get via this way into non-clinical development, but they also, once they get into the competence stream of clinical development, they can grow into clinical development and regulatory as well. But clinical development, I must say, is more or less reserved for medical doctors, with exception of clinical pharmacokinetics. So, well, there are several ways on how you can climb the managerial, the ladder, the career ladder in pharmaceutical industry, and that also the same thing for chemical industries. You can either choose to follow the managerial ladder, or you can stick to your science, become an excellent scientist, and then climb into the scientific ladder. In the managerial ladder, normally you start as a supervisor, and you can grow into a manager, senior director, senior director, and vice president. What is more or less important to climb the managerial ladder, that is when you will have to take on other responsibilities than the typical sciences you are in charge of, like, for example, in interpersonal skills, but also budget management, human resources management, etc. And these are things that you have like to do, but you need also to have the skills of a leader you need to have leadership skills to be able to grow into that ladder. If you are a very good scientist and you don't like to be interested in human resources or budget management, you can stick to the science. And even for very sciences that are very important for the company sometimes wants you to stay in that science because of your reputation and because of your skills and contributions to the company. But then you can do, you can follow a scientific ladder. That's from scientist to principal scientist to fellow, senior fellow, and distinguished fellow. And let's not forget in the pharmaceutical industry, distinguished fellow has got the same level as vice president in a managerial ladder. You will have to know, of course, that when you want to move in the scientific ladder from the one level to the other, you will be actually evaluated very thoroughly by your peer scientists to be able to move to next level. So a couple of tips for a career in pharmaceutical, a successful career in pharmaceutical industry. So I'll just name them: team spirit, leadership, open mind for other ideas, think out of the box, good communication skills, and if possible in several languages. Always be critical for yourself. And if you commit an error, please admit error and correct for it. Get interested in other disciplines in drug research and development. That is the way how we can grow on a corporate level from a toxicologist into a non-clinical development team leader, etc. Flexibility is key. You know, if you're working for an industry, it may well be that for financial reasons or other reasons or competition reasons, you know, that your compound you have been working on so hard that development will be stopped. So in that case, you'll be invited by the company to shift to another project. And, you know, that type of flexibility is key for making a career in pharmaceutical industry. Of course, continuous learning, which you already heard from other speakers, open for change and migrate to other disciplines. You can migrate, you know, along your career line from, you know, bioanalysis into metabolism, but even then to toxicology, and then go further into clinical operations whatsoever. Learn from mistakes. Take on new challenges. Don't be afraid, you know, to jump into some different discipline and learn from it. Change jobs from time to time. Change companies, because if you stay with the same company, you are always busy with your same compounds, and since drug development takes a long time to get to marketing, you know then your toxicology of your drug so good that it becomes boring. So it's from time to time, if you know the toxicology of your whole portfolio and you want to learn more, then well, change companies, then you learn other drugs and other challenges. And that only actually expands your basis of experience. Spend some time abroad. If your company is a multinational and they invite you, for example, to move to the States or to different companies, please take on these opportunities, because then you learn to work with other types of people, you learn to work with other philosophies in working together. And that is really very important in order to have a broad basis of experience. And of course, you know, in the same trend, take international positions if you can. So that gives you a lot of. Okay, to conclude, there are a couple of tips that I find personally very important. First, it's better to stay employable than to stay employed. So if you've got a good job, they pay you well, but it's getting boring and you don't learn anything anymore, well, change jobs so that, you know, you learn and you continue learning. So then you stay employable. You need to be ambitious in doing a very good job. And if you got a good company working for, the rest will follow. People will appreciate very much your efforts and they will open you the way for a career. Don't try climbing the career ladder too fast. Some people, you know, are so ambitious and are so impatient to get at higher levels. Don't do that too fast, because you've got to come to a level where you actually cannot cope with the job. That puts you into a crisis, and all of a sudden, you know, you will be not so much appreciated by the company. And for the last type of suggestion, you spend at about 1/3 of your life on the job. So enjoy yourself and continue enjoying yourself. It's only by that way that you will be creative and you will be really contributing to the company, and you will have a lot of satisfaction of your job. Thank you very much. Any questions?
Thank you. Thank you very much, Mark. Another very interesting, very concrete, very useful presentation. I think that your tips are music for the ears of the EPSA students, because I know that actually they've been working very hard on transferable skills, and this is something which probably the training at our faculties also should improve. So we should not focus only on, hard skills, but also try to prepare our young people, for the job market, improving the soft skills. And also, of course, I also fully agree with the golden rules, even if sometimes, in some countries, unfortunately, it's not so easy to change job very easily. But of course, I think it's a good idea to try to be mobile, and especially, international mobility, that is music for my ears, because I try always to encourage my own students in Rome, to go abroad, to go somewhere else for a while. And then, of course, it is our responsibility to try to encourage them and help them to come back, as soon as possible. So we really apologize, it is late, but indeed all presentations were so interesting that I think, it was worthwhile to leave the time to all speakers for these presentations. We still have more than 140 people online, so this is really, I think, amazing. And this is a confirmation for me that, students, they stay online in webinars when we speak about something that is interesting for them, and of course they leave earlier when we speak about something else that is not so interesting for them. So I think this is a really good result. Thank you very much. So we only have a few minutes for discussion now, but I remind you again, as it was written on the chat, that there will be another webinar. The second part of this webinar will be on December 14th. So I really encourage you to be online again at the same time, 10 a.m. Central European time on December 14th, to talk about some of the positions which were presented by Professor Thomas. But then, of course, there was no time in this webinar to go, and to go deeper on those concepts. But let's take a few minutes for discussion. One question that was actually not coming this time in the chat, but it comes very often when we organize these events. It is related to the PhD. This was actually touched upon by Professor Thomas in his presentation. After a master's degree in pharmacy or in biotechnology, of course, you have different choices. You can take a PhD, you can take in many countries, there are masters, professional masters available, 1 or 2 years. There are specialization schools. There are other, programs which are not exactly PhDs, but, sometimes they are very much focused on some of the activities of the biotech and pharma sector. Of course, let me anticipate that, I think everyone would agree that if you would like to be involved in, research and development, either in academia or in a company, I think a PhD is a must nowadays. You cannot think of becoming head of a lab, I think, anywhere, or head of a department anywhere without a PhD. But the question is more complex, I would say, for the other sectors. So I would like to ask very briefly, we don't have much time, our speakers, maybe starting with Cristina, but maybe also, of course, Mark, and I think Joshi already mentioned that. Which is your view on that? In sectors, not R&D, in the pharma and biotech, how much a PhD is valued, or is it better maybe for a graduate to try to enter immediately in a company and then get the preparation, let's say by working or taking other courses? Can you briefly comment on that? Cristina.
Hi, I can comment that, of course, if you want to stay in research on the early phase, to have a PhD is almost a must, at least if you wanted to have a career that can evolve rapidly, let's say. You can do a nice career also without a PhD, but it will take definitely much longer. What I would advise is that when we think about a PhD, think very well in which area, in which project you want to spend 2-3 years. What I would not advise is to take a 5-6 years long PhD. I don't think it's necessary.
But I mean, Cristina, sorry to interrupt, but I mean, I'm talking about non-research sectors.
Yeah.
Because of course, if you do research, I assume
Yes.
any good company nowadays would assume a person with a PhD or will encourage, an employee to do the PhD after work, which is also possible. There are also the so-called industrial PhDs. So it is possible to do a PhD also in a company, of course, in many countries. But let's say AstraZeneca or other companies, that the speakers of this panel maybe are, knowledgeable about. What is your impression? I mean, would it be better to do a PhD or not?
I mean, I talk from the early phase and research mainly, but I know that in late phase, many people apply directly with the master and then they go ahead and they have a very good career anyway. So, because I think it's more important, the expertise at that point and be able to be broad and maybe also use a secondment, a possibility to spend some time inside your role, but you go in secondment in other department, in other function. So you build your broad experience in that way once you have a job. I don't think the PhD is so important in other functions.
And Mark, would you briefly comment on that?
Well, I'm talking more or less from, a toxicologist's point of view, where you need PhDs, because you need actually people who learn on how to identify problems, how to design projects, and how to come to a final result in the laboratory, etc. But, for example, a typical function in pharmaceutical development, which does not require a PhD, is actually regulatory. There are a lot of pharmacists who make a very successful career in the regulatory arena, and regulatory more or less, let's say, into the clinical area rather than in the non-clinical area. So there are possibilities. There are also possibilities for project management, for which you don't require a PhD. The type of skills that are needed are interpersonal skills, adaptive skills, of course, learning skills, communication skills, etc. And this is more or less some kind of a natural selection process. People who make the best careers are the best. It's as simple as this. So even in drug development, you have different disciplines where no PhD actually is needed. And a typical one is the regular.
Luciano, certainly the contract research organizations, they tend to favor graduates and master's students. I'm not saying there are not people with PhDs in the contract research organizations, but most of their employees tend to be graduate stroke master's students.
Thank you. And Jos, maybe?
Yeah, I gave my opinion on it. In what I call the lab functions, it is essential to have a PhD to make a career, at least. And in the other functions, the office functions, as I call them, it is a competition. If you have a PhD, it is a plus for senior positions and your future career. But if you have not, and if you have other diversifications in your specialization or second diploma, you will have equal opportunities to make a career. So it is not a prerequisite in these functions, certainly.
Thank you very much. So we only have a few minutes, and unfortunately, we had several questions. We cannot address all of them. I hope in the other webinar, as I mentioned, maybe we will have a little bit more time. Another question which came out at the very beginning of this webinar is also very interesting and close to my heart, because UNICA at the moment has several countries not belonging to the European Union. And for some of the students in these countries, it's not so easy to actually travel and go and work abroad like the students in the European Union. and in Schengen space, I would say in general. And I just would like to remind how lucky we are, I mean, in the countries belonging to the Schengen space, because we can move so easily without any problem. So how would you maybe again comment on that? Let's say a student coming from another country that, you know, maybe will require a visa to work in the European Union. How would you, I mean, what kind of advice would you give to, again, to a recent graduate willing to work in the European Union from a country, you know, from which, again, they would need a visa to be in the EU? I don't know if anyone would like to. I know it's not an easy question, but, you know, to start with, if you have any tip, this was a question which came out in the beginning of the webinar.
Maybe one tip can be only to be aware that this can take a very long time, so not to apply at the last moment and so on, because we had really cases in which unfortunately students could not come over just because they could not get the permission. Yeah, okay. So it's a process that sometimes in some countries takes a long time.
Other comments? You know, I
think it's going to be it's going to be a country by country basis. That some countries, their visa application process is easier than other countries, and therefore the students need to, if they're outside the European Union, need to think about it in terms of the country they're going to go into. And I take Cristina's comments on board. Yeah, start it well in advance. Don't leave it till the last minute.
Thank you. And I mean, I can add that there are some European programs such as the Erasmus Plus traineeship program, for instance, which is open to many students of countries which are associated to European program, or so that could be also like a door that they can open, because after the traineeship, maybe they can you know, get to know the environment, and the company also can get to know the student, so that can also help to facilitate the employment. Okay, so I think it's really late. We have to close.
Maybe Luciano?
Yes, please, Chris.
I could also add maybe one thing more to this discussion, and that is that it all also depends quite a lot on having a strong curriculum. And what I've learned also from the student presentation today is that that students are aware that a strong curriculum is not only a curriculum with high grades, but I have tried to add that it is a curriculum developed with a certain vision towards a certain domain like pharma biotech. So what I can add to this discussion is that my impression is that, for example, umbrella organizations like Flanders Bio in the part where I live are really, they are approachable for a strong curriculum. If a student in, let's say, Ukraine or in Romania has a strong curriculum developed not only by high grades, but by a vision, then these people can upload or send their curriculum to possible employers. And then they will have a listening ear and eyes that can help them. Because if you have a good contact with a possible employer, then visa application and all the things that follow will become a little bit less hard and less difficult. That was my my addition to this discussion.
Maybe before closing, I would like to give the floor very briefly first to Jos, if you agree, to the students first, and then to Jos, and then I will close. So Joseph and Andrea, maybe if you can just give us a few comments at the end.
Okay, I can. From the student perspective, I think it was really, in general, all the presentation have been really interesting for students, and it's good to have the opportunities to see from professionals as you are, how you see the current job market and the opportunities that pharmacy students and biotech students have when they are graduating. I see that we all realize that there's a bit of change needed because students have a bit different expectations. That's what is seen by professionals so far. And I think together we can work on delivering a better education, but also the education after graduation through trainings or graduate programs, as was mentioned, for example, with the example of the AstraZeneca and their graduate program, to kickstart the career of young professionals so they can fulfill their potential and be the professionals that they are usually expecting to be at the beginning of their studies.
Perfect. Thank you very much again to EPSA, which partnered so well with UNICA in this organization. And now, Jos, please.
Well, I think it was an interesting morning. What I can add, as I am teaching at the University of Leuven in the Faculty of Pharmacy, and I'm teaching drug development, clinical drug development, and that is in Leuven. Since a number of years, we have now 2 sections in the Master of Pharmaceutical Sciences. The first one is the classic one, and we called it now Master in Pharmaceutical Care. And that is to prepare people for community pharmacies and for hospital pharmacies, but that is a specialization in hospital pharmacy and clinical pharmacy. But then the second one is a Master in Drug Development, and that is to prepare people for the pharmaceutical industry. And then in the first master, for instance, in the first semester, we teach them the whole drug life cycle, from drug discovery, non-clinical development, pharmaceutical development, clinical development, to commercialization. And that is taught by different people with experience in the pharmaceutical industry or at the agency. And in parallel, there is a modular, during the whole semester, a modular teaching in regulatory affairs and in quality assurance. So in the first semester, they get a complete overview of the possibilities or the processes of drug life cycle and the different support functions that go with it, from specialists, all teaching as visiting professors or guest lecturers, as I am doing. And in the second semester, then they have to choose. They have to choose whether they go for focusing on discovery research, either focusing on non-clinical development, focusing on clinical development, or focusing on commercialization. And again, they will have teaching opportunities by specialists. And that is in most of these courses are in English. The first semester it is in Dutch because we're at the Dutch University of the KU Leuven, but the second one is in English. And I'm teaching there, for instance, advanced clinical development. So that is a master's degree that is already preparing people for the pharmaceutical industry. And the masters in pharmaceutical care have a 6 months internship in a pharmacy or in a hospital pharmacy. And the masters in drug development have a 6-month internship in the industry or at one of the agencies. And that is, I think, a good evolution. And I wished it to see it at other universities as well. And probably it is already the fact.
Thank you very much. This is also very useful. I mean, high quality professional masters, I think, are also a solution. And I really hope that, all universities will develop this kind of, programs. So it's really very late. I would like to thank, first of all the participants. There are still more than 100 online. So it was really a great pleasure to have you online. Thank you very much to all the speakers for the excellent presentations, very useful. And I hope to see all the participants again in the next event. It was sent in the chat already. On the 10th of December, we have a high-level event on drug development, including also the EMA, the European Medicine Agency, as partner. And again, of course, together with the EPSA, the European Pharmaceutical Students. And then on the 14th of December at 10 a.m., we will have the second part of this webinar. So we should see you very soon, and have a good week, and see you soon. Bye-bye.